Lonsdale? Yes, Andrew Lonsdale, who is going to present on work that he's, I think was inspired by his time coming to PyCon AU, and was also a Josh submission this year on avocado, cheese, grape, tomato, how I use Python to stop worrying and love Emoji and Bioinformatics, which I'm personally very excited for as a biologist who's come into Python and spent a lot of time looking at AT, Cs, and Gs. So I'll let him get to it.
Thank you. I'm sure you've all got questions. These are some of them, I expect. The biggest one just being why. But let's go through some of them as quickly as we can. So who am I? That's the blurb that's in the PyCon program. But the important bit is that I did software engineering and then decided to go back and study bioinformatics, and it remains the greatest career decision I have made in my entire life. As Adam said, I was at PyCon a few years ago. It's my first time back in 7 or so years, so it's good to be here amongst the community. And I'm known for my loving side projects, and that's what I spoke about last time. So what is bioinformatics? For those who aren't aware, this is an incorrect definition, but it's kind of right about using the science of understanding biological information in particular, or for example, the genetic information that's contained in DNA, and it combines those three disciplines of biology, computer science and maths. There's lots of different kinds of biological information, and I'm going to focus today on the one that I work in on a day-to-day basis, which is DNA and RNA sequences. A quick primer just to recap, if anyone's remembers some sort of high school biology. I'm going to talk about the genome and the transcriptome. The genome is all the DNA contained in our cell, that's in every cell, and the transcriptome is the readout of that genome as the DNA is transcribed from DNA into RNA. And in bioinformatics, a lot of it ends in the analysis pipeline in this red box here. So there's some library preparation, DNA or RNA is pooled and then it's sequenced, so typically there's fluorescence that happens, and the fluorescent signals are then converted into the 4 DNA bases, A, Cs, Ts, and Gs, and that's what you do your analysis in. That's kind of what's called NGS, but there was a big breakthrough. There are newer technologies such as long reads, and rather than fluorescence, in this we have the DNA and the RNA going through a pore in the machine. And as it goes through the pore, the electrical difference is turned into signal, and that signal is then converted into A, Cs, Ts, and Gs, typical with machine learning models nowadays. Regardless of the technique, either way, it's a lot of data. Been a very rewarding career, very interesting problems, but the question you might be asking is, if it's so good, why am I worrying? And the answer is, we have the worst file formats. And that's just not a meme, that's in papers. So what's happened in bioinformatics is that ad hoc things that humans were meant to read have become the de facto standard over the last 20, 30 years, and it just keeps on happening. This is bioconvert, which lets you convert between formats, and the coloured nodes are the ones that most things can convert to, so they're kind of the more basic file formats, and I'll talk about a few of them today that are highlighted there. And typically when you go to learn bioinformatics, you'll learn the biology and the technology to measure it, and then look at the data formats. But this is a Python conference, and we're all data people, so we're just going to look at the data and work backwards from there. So here's a bunch of bioinformatics files in our folder, and we'll have a look at them. This is the FASTA file, it's the most sort of basic one. What it has is sequence information, and then A, Cs, Ts, and Gs. So these are all bunches of DNA sequence. If you don't know what it is, you put an N in there, and each header line starts with a greater than symbol, and you put information about the organism it came from or the machine. Sometimes you might be interested in what are all the sequence headers. And so you might just look for all the lines that start with a greater than symbol. And that'd be an error. And so I've overwritten my file because I've redirected standard output into the file. We've all done it once, all right? And after that, you will use quotes till the cows come home. BioPython saves us a little bit. So BioPython is an old package that deals naturally with faster sequences. It has functions to translate things, and it can read them in SKBio is a bit more recent. It's kind of designed to work more with the scikit family, but they all do the same thing, but they make different design choices, but how they work. So that's DNA sequences. Protein sequences are also in a FASTA file, and so that probably is a different format, so you can probably tell the difference. No, they're the same. Proteins have more individual parts. They have amino acids, so there's more letters in there, but it's still called FASTA files. You know, that could be either. It could be an unlikely protein. It's probably DNA, but there's nothing really to tell you which one it is because it's just very basic text format. The FASTQE format is a bit more complicated. I'll look at this file now. And so this has a header line as well, you can see there. And it's got some ASCs, Ts, and Gs. But the other thing it's got there is all these ASCII characters down the bottom. This is what's called the R1 read. There's also a pair R2. And these are two bits of information that have come from the same molecule, just sequenced from either end. And so we can, if we can't capture the whole molecule, we can get either end and sort of work out where they are in the genome. So the FASTQE format is relatively straightforward. It's got 4 lines. The first line starts with an @ The second line has those raw sequences, ACs, Ts, and Gs. The third has a plus, and optionally some other things. And the fourth line has those quality values, and must have the same number as the sequences. The force field could also actually start with a plus, or an @ actually. And they could be on multiple lines, sometimes. So it's easy to pass, right? Those files are used a lot in quality checks. So modern technology, as you saw before with the squiggle, it's reading ASCs, Ts, and Gs, but no technology is perfect. And so there's going to be errors, and the machine should give you a probability or an estimate of how correct each call is. And often what you do in QC is go through and remove reads that are of very low quality. The encoding there is a little complicated, so I'll just go through it quickly. So as I said, it's the probability that a call is right or wrong. It's called the Fred score, or Q here, and it's the -10 log score of the probability that it was correct. And so what the FASTQ format does is it takes that Fred score and just encodes it as an ASCII value. So the bad characters down there are punctuation, all up to good characters there. Again, BioPython to the rescue. It can read in those file formats, and then you've got an object. You can extract the qualities and the reads if you want. And as you can see here, you can get them in the numerical format, which is what it's intended for. So that's why I worry a bit. But as I say, Python has kind of makes bioinformatics a bit easier. And so what you do in bioinformatics is often, I said, you do quality check, and this is FastQC. It's kind of the de facto standard to look at the quality of those FastQ files. It'll tell you in each position for all the reads that you've got from some experiment, what is that quality score. And the green zone there above 30 is pretty good quality. And typical of the machines, the quality degrades as you get towards the end. And so it goes down there. So that's FastQC, and that's FastQE, which does the same thing, but with emoji. And it's much better. You can see the quality reads start good, and then they get shit towards the end, which is what you want to teach when you teach people. So that -Q log score, that's how it maps it. So you can see that the probability is quite low of an error, but when the probability is high of an error, again, you get the poo emoji, and it's on the log scale. So that's what it looks like when you plot it on the log scale. So I think that's more interpretable than those ASCII characters. FastQA lets you map all the characters, and you can choose which ones you want to use for it, and it's kind of useful. So that's what it does. The question you're probably really asking is, but why does this even exist? So the last time I was here was 2017. I said I talked about side projects, and actually it was in a parallel session. So I was talking about side projects. There was a bioinformatics talk on the other room, and something about Unicode on the other room. And I wish I could have gone to the other talks that I was in. And by the end of my talk, my audience might feel the same way. So what could I do? I did one more thing. And so at the very end of my talk, I talked about how we encode these qualities in ASCII values, but that's pretty arbitrary, right? So why not we encode those values with emoji? And the very, very last slide, what I did was I said, well, this is a typical example, the equivalent of FastQC. Let's just summarize it there. And that really should have been the end of it. I started getting support requests after I put it on GitHub. people find on Reddit periodically, tell my friend I love him, you know. And I was like, someone found it and put it on what we call X nowadays. And I was like, oh yeah, we could use that for education, that'd be great. And my friend's like, no, call the side project police. Andrew, finish your PhD. And look, I resisted for about a year. And then 2017 PyCon, there was a free slot in the lightning talks. So what was I to do? So I expanded it a bit more. Instead of just the mean score, we looked up the maximum and the minimum so we can get the whiskers of those box plots that are on there. And, well, after that, it was hard to resist developing further. But I got there in the end, right? And ever since then, I've worked on it. The design is very basic, actually. Let's take the FASTQE format, pass through it, let's calculate those summary statistics, and then convert them into emoji. Under the hood in Python, it's just Python to take ASCII characters, turn them into integers, summarize them, turn them back into ASCII characters, and then map them to emoji. So it's really obvious to people who are learning the FASTQE format how you get from emoji to the scores. It has what I like to call a presentation-driven development. So I find a dubious link in some talk that's coming up and use that to justify doing it. And you can see from the code frequency that it leads to features about once a year. For that lightning talk, I put it in the minimum maximum. I got it ready for PyPI. Had a lab meeting, I put it on Bioconda. I had another talk, I got it to work on compressed files. Then people started using it in formal education. So in the US, it's been used in undergraduate courses, and you can see the effect of quality trimming in the middle there, but you can see it much clearer from the top in emoji, and when you do some quality checking, suddenly the shit emojis are gone. And then it kept going. So 2020 was a tough year for everybody, but there was an online conference that combined Bioinformatics open source software and Galaxy. And Galaxy is a great web interface that gives access to a lot of bioinformatics tools, a lot of them written in Python and compute. And in that talk, actually, I gave a talk called Please Contribute to FASTQE so I don't have to, because I really wanted people to help me out. And actually, I was in a parallel session with that as well. And so when I have a parallel session, what I tend to do is try and show off. And so what I did was I said, could we get FASTQE to work in Galaxy? And that could have been the end of that as well. But at BOSC, they do a hackathon, much like the Code Fest here that will happen on Monday, Tuesday. And so over the course of the days, people really worked on integrating the tool into this Galaxy platform. It then got on their training network. And so every year they have a smorgasbord or a training academy where thousands of people across the world learn bioinformatics. And it's in there. I've recorded a video for them about how it works in there. Now, the trick is that actually it doesn't do much. FASTQE just prints the command line. They've written a nice HTML interface that just uses sed to filter the output and create a HTML web page, and it looks like that instead of on the command line. Of course, it wasn't perfect, so it doesn't quite work with the latest version. And in their tutorials, they made it clear that my tool works on the short reads, but not the long reads with that squiggle that I showed you before. And I took that personally. So we added a HTML report to FASTQE, so they could work with the long reads. And so now we can window over very long reads and preview what's happening in the actual reads. And so now in Galaxy, if you can go in and use FASTQE there, you get the interactive report, and you can actually see the reads and really inspect them. And look, that's not just Galaxy. So MultiQC is a Python tool for aggregating bioinformatics reports, and the HTML output works on that. So, is anybody using it? Yes, I think so. So we've got 27,000 downloads on PyPI, and Bioconda there's also another 10,000. And you know it's not the most popular package in the world, but it's also not the least popular package in the world. This is sort of the current state of the command line. So it does a lot of things. You can use custom emoji. As I said, you can collapse the min. You can do a window, do the minimum and the maximum. And that's sort of the state of FASTQE. Here we are in 2024. So there's examples of use of it in education. It's informal, accessible. I want to finish that report and release it. And it's sort of going pretty well. So we've answered a couple of questions there, I think. But in 2020, when we had that Code Fest and really worked on getting it into Galaxy, there was another major outcome, which was back to that original PyCon demo. And instead of summarising the quality, what if we just converted the files? And so putting the emojis in the actual reads rather than summarising them. And it kind of brings up an interesting question, which is, why does DNA need to be stored in English? This is the famous treachery of the images. That's not a pipe. That's not a nucleotide. That's a picture of a nucleotide. Neither are these. We use the first letter of the chemical name to represent them, but that's not the nucleotide. They're just letters. So in bioinformatics, we're stuck on these text-based formats. Increasingly, we're using binary formats, but it's a slow and hard slog. And there's lots of time wasted compressing the text, decompressing the text. And so those encodings are really essentially arbitrary. So why not use corn, apple, tomato, grapes? And so after that, it was hard to concentrate in FASTQE because we had Biomojify. So Biomojify attempts to resolve this by addressing some of those file formats. So from the command line, you run it. You use a subparser to decide what kind of format you're going to look at, and then you can convert. So looking at all those files we looked at earlier, the FASTA file, there becomes a lot nicer there with the emoji you choose. The protein file, you can clearly tell it's different here. We're using all these different emojis to represent the different amino acids in the protein. If you're feeling festive, you can use a custom dictionary. So for Christmas, Christmasify your DNA. And the FASTQE format, we can combine those things. So we can change the nucleotides, and we can change the quality strings as well. Some of those other formats, so the variant VCF is about recording the changes in DNA against some reference. You can sort of say, how are you different to the reference population? And that's, again, written in text, allegedly human readable, but there's not much point to it. You usually use much more advanced tools. So Biomojify will tell you what's the reference, an avocado, and what you have, or you've got an avocado and a corn, and was that read passing the quality filter, yes or no? So, a little bit tongue-in-cheek, but could we make better file formats? You wouldn't get any clobbering if you had an emoji in there when you were looking at a FASTA file. If we're going to break compatibility, we may as well go for it. So I'm really thinking like some crazy proof of concept to say, this is how you could do the best version of a bioinformatics file format if you wanted to use an emoji. So that's the state of Biomojify in 2024. Works best on Linux and Mac. No one's really using it educationally I know of, but I want to change that. And as I said, I really want to get that on the Galaxy platform as well. So I hope I've answered the why question. And really, for me, I think education is a pillar of research, and education works best when it's engaging. And so I hope that when undergraduates and new people to bioinformatics use these tools, they really do find it engaging and they are more interested in the biology and the technology underneath and get to know things. And personally, as a scientist, I code infrequently, and so it's a great excuse for me to try new Python tricks and to keep my skills up. And basically, I just can't help myself from working on this. So I hope I've answered all those questions. These are all people that supported FASTQE and Biomojify going through it. And I also thank Alicia for indulging me whenever I say I want to go and do this away from my day job at Peter Mac. So that's all the questions. I could do one more thing, and I was going to do one more thing, but then I thought twice about it. Because I want to talk about a thing that I learned about at PyCon, which is the bus factor. And for those that don't know, the bus factor is how many people would need to be run over by a bus for your project to end. And, for me, the bus factor of these projects is 1. And the one more thing kind of tricks, that's what got me here in the first place. It's very sporadic development, but it's never quite got off the ground and been used to the extent that I wanted. So the code for these tools is a mess. I'd really appreciate help and people getting involved. I think that there's lots of open issues to get involved, and I'm open to any kind of suggestions. These are all the issues there. I mean, Python 3.13 is causing havoc with the BioPython system, and so we're pinned to 3.12 for the moment. And lots of ideas, and there's a few bugs and enhancements and those kinds of things. As I said, I learned about bus factors here, and I moved it from my own repository to create an organisation for these tools, and so the infrastructure is kind of there. I mean, the Galaxy wrapper that I showed, there's just JavaScript banged right into the Python code. It is ugly. There's CDN network URLs that are just in there that will change at some point. So I really want to think about templating, and these are some of the template libraries I've looked at, but it's one of those things I'd really appreciate anyone's interested to come and talk to me about those. Almost all the development for these tools happens in the hours before I'm about to talk about the project. So I would love contributors to let's spread the load and really make these projects a bit more sustainable. Thank you. Oh, was that only 20 minutes? Oh, all right. I got one more thing. All right, hang on. So, the SAM-BAM format aligns reads to a reference. And so it's another one of these text-based ones. There's lots of human references, HG19, HG38. For this audience, HG19 is the Python 2.7 of human genome references. So if you don't know anything about bioinformatics, now you know. If someone says it's done in HG19, send it back. All right? So this is what the format looks like. And again, it's pretty incomprehensible. It's not meant to be for a human, so it should be binary. But we're in this weird space where everything's text just for reasons. This is still a work in progress because there's things like this cigar string which kind of have different combinations of how well did this read match the reference genome, and working out a nice emoji representation of this is more time than I had this morning. So that's still working, but that kind of works. And so Biomojify, at the moment, can almost pass these SAM files and convert that. So that's something I'd like to work on. And, well, we've still got time. All right. So one more thing after that. Do we? All right. Oh, this is a good one, actually. I'll do this one. This is quick, because I really want help on this one. So it's very hard for people to use Biomojify FASTQE if they're not actually savvy with the command line. So I'd really love a way for people to use it. Looked at Python Notebook, so you can run it in the notebook and teach it that way. I've dipped my toes in, but the HTML report doesn't really work, so we can't do that. I've tried Pyodide to use WebAssembly, and it sort of works, but I just don't quite have the skills to do it, and I couldn't get it. So for the moment, for accessibility of these tools, the only way to do them was to just totally rewrite it in Rust. So that's Rust FastQE. That kind of works. But I don't really want the Rust version to be the final version that sort of exists out there. I'd really like it to sort of come back to Python, and I'm looking at ways to how to integrate the Python and the Rust components so they work together. So if anyone is really into WASM and Python, please get in touch, because I'd love to talk about that. All right, I'll stop there.